A-Level Biology 9700, Paper 4, Variant 2, May June 2025. And students, in this video, we will discuss the question number 1 and question number 2. So question number 1 is, figure 1.1 is a diagram of part of the inner membrane of a mitochondrion. And in this figure 1.1, they have shown the 4 carriers. And question A, part 1, the coenzymes NAD and FAD deliver hydrogen atoms to the electron transport chain. So, with reference to figure 1.1, state which of the 4 carriers receives hydrogen atoms from the reduced FAD. So, there they have shown the 4 carriers which are 1, 2, 3, and 4. So these 4 protein complexes are embedded in the inner mitochondrial membrane. So this is the inner mitochondrial membrane, or 4 of them are embedded into it. So the process involves the delivery of hydrogen atoms. So the hydrogen atoms split up into protons and electrons during the process. Hydrogen atoms, they split that into electrons and protons. So during this process, what happens that reduced FAD and reduced NAD, you can see over here, they delivers its in this the reduced FAD, it will deliver its hydrogen atoms, electron and protons, to a specific carrier in the electron transport chain. So reduced NAD deliver its electrons to carrier 1. Carrier reduced, they are 2, that is the reduced NAD, and other is reduced FAD. So the reduced NAD, it delivers its electrons to carrier 1. And now we have to write the FAD, about FAD. So the reduced FAD deliver its electrons to carrier 2. So carrier 2, which is succinate dehydrogenase, or complex 2. We say that succinate dehydrogenase, or 2. So we will write over answer. It is carrier 2. Now the next question is, the hydrogen atoms that are delivered to the electron transport chain by reduced NAD and reduced FAD, they split up into electrons and protons. Already told you. And during this splitting up of these into electrons and protons, the energy is released as electron pass along the electron transport chain. So describe the events that occur as a result of this release of energy. So what happens during this? During this process, the electrons are passed sequentially along the chain of carriers you have seen in the figure 1.1. That is 1, 2, 3, and 4 releasing energy at each step. So the energy released is used up by the complexes 1, 3, and 4 to pump the protons from the matrix into the inner membrane space between the inner and outer membrane. So the buildup of protons in the inner membrane space creates an electrochemical gradient, which is then used by carrier 5 to synthesize ATP through chemiosmosis. So these are the you can say events that occur as a result of this release of energy. So I would like to write the released energy is used up. to pump electrons and protons from the matrix into the inner and outer membranes. Inter and the second event can be the buildup of protons in the intermembrane space creates an electrochemical gradient which is then used by carrier carrier 5 to synthesize ATP by or through chemiosmosis. So it was part 2 of the question 1. Now move towards the next question, which is describe how structure 5 in figure 1.1 is involved in oxidative phosphorylation. So here in this question, we have to talk about the involvement of the structure 5 in. So first of all, in this, I would like to say that it is crucial. So structure 5 is the enzyme synthesis, ATP synthesis, and it is crucial to the final stage of oxidative phosphorylation called chemiosmosis. So this ATP synthesis is involved in this synthesis in synthesizing ATP. So proton gradient, the preceding carriers 1, 3, 4 chain use the energy released by electrons to actively pump protons from the mitochondrial matrix into the intermembrane space. So this creates a high concentration of protons in the inner membrane space and low concentration in the matrix, establishing an electrochemical gradient known as the proton motive force. So as a result of this, the ATP synthesis is a channel protein that spans the inner mitochondrial membrane. So this accumulates proton cannot diffuse back across the membrane except through this channel. So ATP synthesis helps to protons flow back down their electrochemical gradient from the inner intermembrane space back into the matrix through this. So the movement of hydrogen ions provides the mechanical energy to power the enzyme's catalytic sites. So ATP synthesis uses this energy to catalyze the reaction. So I would like to say that the flow of protons during this photophosphorylation, the ATP synthesis act as a molecular machine that converts the potential energy stored in the proton gradient into the chemical energy stored in the phosphate bond of 84. You can say, uh, after this description, I think I would write all these points which I have told you. So I would like to write this. So the preceding carriers of the electron transport chain 1, 3, and 4 of use the energy released by electrons to actively pump protons from the mitochondrial matrix into the inter intermembrane space. Intermembrane space. This creates a high concentration of protons in intermembrane space. And a low concentration in matrix. Establishing electrochemical gradient. The accumulated protons cannot diffuse back across the membrane except through this channel. Then protons flow back down their electrochemical gradient from intermembrane space back into the matrix. Through it. So the movement of protons or hydrogen ions provide provides mechanical energy. Mechanical energy to power the enzymes catalytic sites. So the reaction is catalyzed. Moving towards the next part of this question. This question is, cyanide ions are highly toxic. Cyanide ions bind to carrier 4 in figure 1.1 and inactivate the carrier. So suggest and explain how the binding of cyanide ions to carrier 4 can have an effect on respiration. So the question asks us to explain the effect of cyanide ions binding to carrier 4 on respiration. So carrier 4 is typically the final enzyme complex in the electron transport chain known as cytochrome oxidase. So carrier 4 is cytochrome oxidase. Cytooxidase. So this is cytochrome oxidase. So carrier 4, you can say it will affect the of cyanide on respiration. So cyanide is the potent inhibitor because it directly interferes with the final step of the electron transport chain in aerobic respiration. So how do it will affect? So cyanide ions bind irreversibly to carrier 4, which is located on the inner mitochondrial membrane. So this binding will inactivates or inhibits the enzyme. So when carrier 4 is inhibited, electrons cannot be passed to oxygen. So the flow of electrons to the entire electron transport chain stops. So this prevents the pumping of hydrogen ions or protons from the mitochondrial matrix into the inner intermembrane space. Therefore, the proton gradient across the inner mitochondrial membrane is not maintained, it is destroyed. So the flow of protons back into the matrix stops. As a result, oxidative phosphorylation mechanism for ATP synthesis ceases, leading to a drastic drop in ATP production. Then electron transport carriers cannot pass on their electrons, they remain in their reduced states. So the lack of oxidized coenzymes means that the earlier stages of the respiration, especially the Krebs cycle and link reaction, are also inhibited because they run out of the necessary coenzymes to accept electrons, that is the oxidized. So the cell must then rarely solve on glycolysis for ATP, which is a much less efficient process. So now I'm going to write all this. I will start with the first step, which is that the cyanide ions binds ions bind irreversibly to carrier 4, which is cytochrome oxidase. So, which is located inner inner mitochondria. This binding inactive inactivates or inhibits the enzymes. When carrier 4 is inhibited, electrons cannot electrons cannot be passed oxygen. This prevent pumping of hydrogen ions from the from mitochondrial matrix into the intermembrane space. Since the electron carriers cannot pass on their electrons. They remain in their reduced state the lack of oxidized coenzymes, which are FAD and NAD, reduced FAD and reduced NAD. Means that the earlier stages of respiration, especially the Krebs cycle and Lick reaction are also inhibited. The cell must then rely solely on glycolysis for ATP, which is a much less efficient process. So it was all about the question number 1. Now moving towards the question number 2, and question number 2 is, when organisms reproduce, they pass on their alleles to the next generation. There are many factors that can affect how alleles frequencies change over time in a population. Explain how genetic drift and the founder effect may affect allele frequencies in populations. So we have to discuss about genetic drift and founder effect, which may affect the alleles frequencies in the populations. So first of all, I will start with the genetic drift. So genetic drift is the random fluctuation of allele frequencies from one generation to the next due to the chance events. So in a small population, it is possible that by pure luck, some individuals with specific alleles do not reproduce on their offsprings, are simply not produced, or they do not survive irrespective of their fitness. This random sampling of gametes or individuals can lead to disproportionate representation of alleles in the next generation. So it typically causes alleles to be lost um, or fixed in a population, significantly reducing genetic variation within that population. So genetic drift has a much stronger and faster effect in small populations because the loss or fixation of even a few individual alleles has a larger proportional impact on the overall gene pool. So in this way, it affects. Secondly, the founder effect is a specific form of genetic drift that occurs when a new population is established by a very small number of individuals that are isolated from a much larger parent population. So the small group of founders likely does not carry the same proportion of alleles as the original larger population. By chance, rare alleles from the original population may be overrepresented in the new group, or common alleles may be missing entirely. So the allele frequencies in the new isolated population will immediately and drastically reflect the gene pool of the few founders, not the original population. This often results in a reduced genetical diversity in a new population and a higher incidence of certain alleles so than in the original source population. So it was in this way this genetic drift and founder affects the population. So first of all, I would want to write the about the genetic drift, how it affects. So genetic drift, it is the random fluctuation genetic drift fluctuation of allele frequency. Frequencies from one generation to the next. Due to chance events in a small population, it's possible that by now some individuals with specific alleles do not reproduce or do not survive irrespective of their fitness. This can leads to disproportionate the presentation of alleles in the next generation. It typically causes alleles to be lost. To be lost or fixed in a population, reducing genetic variation. So, genetic drift has stronger and faster effect on populations. On small population. The second founder effect is a specific form of genetic drift that occurs when a new population is established by a very small number of individuals that are isolated from a much larger parent population. So, founder effect special type of genetic drift in which small group is isolated from larger parent population. Which does not carry the same proportions alleles as the original population. So the frequency allele frequency in the new isolated founder population will drastically reflect the reflect the gene pool of new founders. This often results in a reduced genetic diversity in the new population. So it was question number 2. So it was all about the question number 1 and 2. We are going um, to finish this question today. So please like, share, and subscribe my channel to watch more videos on this paper and on other papers. Thanks for watching.